Hello everyone and once again welcome to Bioline. Today we will talk about the lactose operon and this is the second video for the lactose operon. I have already uploaded the first video. So I'll request you all to first go and watch that video and then come to this one for the better understanding. I'll also mention the link of my previous video in the description box. Okay, so for better understanding, all of you please watch that video first and then come to this one. Okay, so in the previous classes we have already talked about that what is a lactose operon and why it is needed in the bacteria. Okay, we have seen that this is a lactose operon which is also known as Lac Operon, a operon which is responsible for making the genes, or we can say for making the enzymes responsible for the lactose operon, or sorry, lactose metabolism. Right, this we have already talked about. We have also seen that this lactose operon can be controlled by two major proteins. The first is the Lac repressor. Okay, first is the Lac repressor, and the mechanism of the Lac repressor we have already talked in the previous video. Today we'll be talking about the second type of protein which will be controlling the lactose operon, that is our cap protein. Okay, which is also known as the positive regulation or the glucose effect on the Lac operon. So let's start. Okay, this is what is the Lac Operon look like. We have already seen that it consists of three structural genes, that is Lac Z, Y and A, and all of these three genes are making the enzyme which is responsible for the lactose metabolism. Okay, then just upstream to that we have the operator region where the Lac repressor binds and it will negatively regulate the operon and it will switch the genes off. Right, this is the promoter region which has a polymerase binding site. Okay, just upstream to that there is another sequence which is known as cap site or cap binding site. Okay, so today we'll talk about this cap site and this cap protein. Okay, this is a cap site where a cap protein along with its another molecule, that is cyclic AMP. Okay, this cap protein along with another molecule called cyclic AMP, it will bound to this cap site and it is going to give the positive effect. Okay, this will promote the binding of RNA polymerase. Okay, all of you remember that that's repressor kya karega, repressor will decrease the binding of the RNA polymerase, right? Uski binding ko wo kam karwaega, and that's why the transcription will go at a very low level. Okay, but this is an activator. Cap is an activator protein, a catabolite activator protein. Okay, and that's why it is positively regulating the Lac operon. That means it is going to enhance the polymerase binding on the promoter side. And how this is going to do, this will cause the bending of the DNA. Okay, this is going to cause the bending of the DNA via which the promoter, which is bound to the RNA polymerase, will have another activators, and then a strong transcription can be done, or we can say the rate of the transcription of these Lac genes will increase. Okay, we'll study more about the cap protein. This is the second regulation that we'll be talking about the Lac operon. Okay, this is a cap protein which is an activator. Cap stands for catabolite activator protein. Okay, the another name for cap is also that is CRP. Why? Because just now I have told you that cap cannot bind to the cap site individually. This is the cap site, you can see here. The cap will first, this cap has to first bound with another small molecule, that is cyclic AMP. Okay, and then this combined structure will go and bind to the cap site, which will have the positive effect on the Lac operon. So because it is also binding, this cap site or this cap protein can bind to the cyclic AMP, it is also known as CRP. CRP stands for cyclic AMP receptor protein, because it is a kind of receptor for cyclic AMP. Okay, because cyclic AMP is allosterically controlling the activity of cap protein. Right, now the gene encoding cap is located elsewhere on the bacterial chromosome, not linked to the Lac gene. Okay, देखिए, the protein cap, this protein cap is not linked to the Lac gene. Okay, the gene responsible to make this protein cap is somewhere else on the chromosome. Okay, but this cap site, that is the site where this cap protein is going to bind, this site. Okay, this is in the, we can say, in the relation, or this is near to the Lac genes. The cap can bind the DNA, and at which point on the DNA it can bind to the cap site. right, and activate the lac genes only in the absence of glucose. Very important. This effect of the scap protein is also known as glucose effect. Why? Because the scap protein will come in the action whenever the glucose is there. Okay, this is in the concern of glucose actually. Fine. So we'll see how in the presence of the glucose the cap protein is bound or it is unbound. We'll talk about it in the another slide. This entire controlling mechanism via the cap is also known as positive control of the Lac Operon, or it is also known as the glucose effect of the Lac Operon. Okay. Now, before we start further about the cap, let us take a condition where a bacteria is kept in a media which contain two sugars. Okay, the first sugar that a bacteria will encounter is the glucose, and also in the same media there is a lactose sugar. So always remember, the preferential source for the energy, the preferential sugar for a bacteria is always the glucose. So if we plot a graph here, okay, you can see here this is the number of cells, that is the bacterial density, and versus time. So you can see if the bacteria is having both the sugar in the media, firstly it will always utilize glucose. Why? This I have already told you, because number one, the very important thing is glucose is a monosaccharide. It can directly enter into the glycolysis. Okay, but the lactose is a disaccharide. First it has to be broken down into the monosaccharide, that is glucose and galactose. It will be broken down into galactose and glucose, and then they are going to enter into the glycolysis. तब ये जाके glycolysis में enter करेगा. So here little bit more energy is required for the breaking of lactose. Okay, but if directly glucose is available, then this energy can be saved by the bacteria, right? And this will directly enter into the glucose. So that's why the preferential source for the source of energy for the bacteria is always glucose. So you can see first it is going to utilize the glucose, and you can see the exponential phase of the growth of the bacteria. Okay, so till this point the bacteria is just utilizing glucose and it is growing, it is increasing in number. Okay, it is dividing and increasing in number. Then what happened? Then the level of the glucose goes down. In this area what happened? Let me change the color. The level of the glucose goes down. Okay, then what happened? There will a very small, or we can say a short lag period will be there. What is there now? So already in the media the glucose has been over, glucose khatam ho chuka hai. Okay, now only lactose is there. Now bacteria do not have any option, it has to utilize lactose as a source of energy. So what happened? The growth of the bacteria will cease for a very short period, and that period is called lag period. So what will happen in the lag period? In the lag period, the bacteria is going to switch on the lac genes. Okay, lac genes will be switched on. Why? Because glucose is now no more. Okay, only lactose is available, and lactose ke metabolism ke liye lac genes are required. Okay, sorry, not switch off, it should be switched on. So the lac genes are switched on. We'll say the Lac Operon has to be switched on. That's why during the lag period, the bacteria is just switching on the Lac Operon so that the lac genes can be transcribed and the product of the lac genes can be available. Okay, and this is also happening because of the leaky transcription. All these things we have already talked in the previous video. So I'll request you all to please go to the previous video. That is the part 1 of Lac Operon for the leaky transcription and the lac genes, the product of lac genes and all. Okay, once the lac gene is transcribed, the product is being there, then what happened? Again, you can see a log phase is there, an exponential phase will be achieved. Okay, वापस से क्या होगा? Exponential phase achieve किया जाएगा and the lactose will be utilized. Right? So now you can see this type of growth where one exponential phase, this is exponential phase 2 are separated by a small lag period. Okay, and this type of growth is known as dioxic growth. इस growth को कहा जाता है dioxic growth. Dioxic coming from the Latin language where auxilium means help. Okay, here because the two sugar are helping the bacteria to grow, that's why this type of growth curve is known as dioxic growth curve. Okay, here one important thing that you should note here is, you can see that when the glucose was there in the media, when glucose was high, I'll just write it here, when the level of glucose was high, the Lac Operon was switched off. Lac Operon क्या था? Was switched off. Right? Lac Operon was off. Why? Because definitely na, when glucose is there, there is no need for the production of the enzymes responsible for the lactose metabolism. राइट क्योंकि ग्लूकोज यूटिलाइज हो रहा है, लैक्टोज के लिए मेटाबोलाइज करने के लिए जीन्स की जरूरत ही नहीं है. दैट्स व्हाई इन द प्रेजेंस ऑफ ग्लूकोज द लैक्टोज operon is switched off, and this is known as catabolite repression. इसे कहा जाता है catabolite repression. That means when the glucose level will go high, the Lac Operon will be switched off. Lac Operon क्या रहेगा? बंद रहेगा, उसको repress कर दिया जाएगा. That's why this is called catabolite repression. Okay, so how does this glucose switch off this? So कैसे ये Lac Operon को बंद कर रहा है? This we will try to understand. This is doing it with the help of a protein called CAP and cyclic AMP के combination से हो रहा है. Let us try to understand. Okay, see, we have already talked that in the absence of glucose, what is happening? See, when there will be no glucose, definitely, देखिए, when there will be glucose, I'll just write it here, when there will be high glucose, okay, the Lac Operon will be switched off. Lac Operon will be off. And when there will be no glucose, the Lac Operon has to be switched on, because there now the lactose has to be utilized. Lactose को utilize करना पड़ेगा. Okay, so when there is no glucose in the media, the bacteria that time they make what? They make cyclic AMP. This cyclic AMP will activate CAP. Okay, and then CAP will switch on the genes and allow the cell to utilize the alternative source, that is the lactose. Okay, what is happening? जब तक ग्लूकोज था, लैक्टोज बंद था. अब जैसे ही इन डायोक्सिक कर्व में हमने देखा है, what we have seen, कि जैसे ही ग्लूकोज का लेवल कम हुआ, okay, as soon as the glucose goes down, what will happen? There is a enzyme called adenylate cyclase that gets active. वो क्या हो जाता है? Active हो जाता है. Right, within the cell. Once the adenyl cyclate get active, it will increase the concentration of cyclic AMP within the cell. Once the cyclic AMP is high, it will go and combine with CAP. Okay, cyclic AMP किसके साथ combine हो जाता है? CAP protein के साथ combine हो जाता है. This entire complex will go and bind on the CAP site. Okay, and this complex will give the positive effect. It will go and do the binding on CAP site. It will go and binding on CAP site. As a result, the Lac Operon will get switched on. The Lac Operon will get positively stimulated and it will be switched on. So this all will happen when the glucose level is low and there is higher lactose. Lactose ज़्यादा हो रहा है, glucose कम है, तो ये पूरी story आपको याद रखनी है. What is happening? Adenyl cyclase, the enzyme, a membrane-bound enzyme, membrane-bound protein, this will get activated. Once it is activated, it will increase the cyclic AMP concentration. How it is going to convert the normal AMP, जो हमारे normal AMP हैं, ये उनको cyclic AMP में convert कर देगा. AMP stands for adenosine monophosphate. Okay, so this will form a cyclic structure and that will form a cyclic AMP. Okay, in cell signaling we have already talked that in the cyclic AMP are the important secondary messengers, right? जो adenyl cyclase जो उनकी activity या उनकी concentration को बढ़ाता है. Okay, so that's why it will be wasteful for the CAP to induce the expression of Lac Operon if the lactose is not present. अगर lactose नहीं है तो definitely CAP क्या करेगा? CAP will not bind to this site. Okay, to understand further, we'll see some other slides. Now, हमने already बात कर ली कि glucose क्या होता है? It can directly enter into the glycolysis without the modification. That's why glucose is the preferential source of energy over any other sugar. Okay, so whenever the glucose is available, the other sugars, the operon or the utilization of the other sugars will be reduced, and this phenomena is known as catabolite repression. This I have already told you in the previous slides, that what is catabolite repression, that in the presence of glucose, the metabolism or the utilization of any other sugar is reduced, and means they will be repressed, the operons will be repressed, and that's why this is called catabolite repression. Okay, now the efficient transcription of a Lac Operon take place when only lactose is present and glucose is absent. ठीक है, डेफिनेटली जब लैक्टोज का कंसेंट्रेशन ज्यादा होगा तभी तो लैक ओपेरॉन को स्विच ऑन करने की जरूरत पड़ेगी ना. Yes or no? Because Lac Operon are the gene will transcribe the genes which is utilizing the lactose as the initial source, right? So we'll say when the lactose is low, or in the another term you can say when the glucose is low. Sorry, when the lactose is high or glucose is low, only in that condition what will happen, the operon will be switched on. Operon तभी on होगा अगर lactose ज़्यादा है. ठीक है, तो let us try to understand this in the further. Now हमने अभी बात की कि whenever the glucose will be less, the intracellular concentration of cyclic AMP will increase. This cyclic AMP is the allosteric effector of cap. Okay, so you remember, if this is the cap site, just now we have already talked about this, that if this is the cap site and this is the cap protein, it cannot individually go and bind here. It can only bind to the cap site when it is in the combination with the cyclic AMP. Why? Because the cyclic AMP is the allosteric effector of this cap protein. Okay, so that's why the another name for cap is also CRP, that is cyclic AMP receptor protein. This cap will bind to this cap site and do the positive regulation of the Lac Operon only when it is bound to cyclic AMP. And just now we have seen cyclic AMP कब bind हो सकता है? Only when its concentration will go high. Right, only in that condition it can go and bind here. And when will the concentration will go high? When the activation of adenylate cyclase, or we can say adenyl cyclase, active हो जाएगा. And when can this be active? It can only be active when there will be low glucose. Glucose का concentration कम होगा. When there will be low glucose, only at that time this condition can be seen. So only when glucose levels are low, the cyclic AMP level will go high. Okay, and the cap will bind to the DNA and it will activate what? The Lac genes, right? Okay. You can see here with the help of a small diagram के through हम यहाँ पे देखते हैं. This is what the promoter is. This is the operator. This is our gene Z, Y, A. ओके, ये प्रमोटर पे आरएनए पॉलीमरेज बाइंड करता है, ऑपरेटर पे रिप्रेसर, विच रिप्रेसर? लैक रिप्रेसर यहाँ पे बाइंड करेगा. ठीक है, एंड इन दिस केस यहाँ देखेंगे आप. ओके, एंड कैप साइट पे कौन बाइंड करता है? साइक्लिक एएमपी along with cap. Okay, cyclic AMP along with cap can bind to the cap site. Okay, these are the regulatory sites and their binding factors. So these are the cyclic AMP and cap will bind here, RNA pol will bind to the promoter, and repressor will bind to the operator. Okay, now the prerequisite for the expression of genes, या कौन से genes की बात हो रही है? लैक genes. कौन कौन सी conditions हैं कि लैक genes तब repress हो सकती है? Okay, only when O site is empty. That means जब operator empty हो, that means there should be no lac repressor. When there is no lac repressor, right, lac repressor नहीं है to repress the activity of the gene, only then it can be activated. दूसरा, when cap is bound, when the cap site is bound to cyclic AMP and cap protein, जब इस पे cap site पे यहाँ पे bind होगा, because it will give which effect? ये positive effect देगा to the lac gene, only then condition it will be activated. Okay, lac repressor यहाँ पे bind नहीं होना चाहिए, because lac repressor क्या है? Repress करता है lac genes को. Okay, that's why it should not be bound for the expression of the gene. Okay, now we'll consider three conditions. A condition where only glucose is there in the media. Okay, we can say this is a media which contain of only glucose, and these are the bacterial cells. Okay, these are the bacterial cells. So under this condition, what will happen? Under this condition, we can see the media has high concentration of glucose. Okay, see, there is always an inverse relationship between the concentration of glucose and cyclic AMP. So when the glucose concentration is high, that means the first thing that should come in your mind is कि now the Lac Operon will have to go off. Lac Operon को अब बंद होना है. Why? Because glucose is high. That's why if there is lactose, then also the Lac Operon's bacteria is not going to utilize it, and the Lac Operon is not needed. Right? उसकी genes का expression की जरूरत नहीं पड़ेगी. And if there is no lactose, even in that condition, the lac genes has to be switched on, because always if there is glucose, the preferential source has to be glucose only. Okay, so under that condition, what will happen? The adenyl cyclase will be inactivated. Once the adenyl cyclase will get inactivated, there will be no cyclic AMP, or we can say low level of cyclic AMP will be there in the cell. Once there is no cyclic AMP or low level, the cap remain unbound. Okay, the cap protein will remain in the form of cap. It will not be bound by cyclic AMP. Okay, and when cyclic AMP is not bound to the cap, we have seen that it cannot bound to the cap site. Okay, this cap cannot bound to the cap site, and because cap is a positive regulator of the Lac Operon, but now cap is not binding to the cap site, that's why the promoter cannot initiate the transcription and the operon is repressed. Operon suppress हो जाता है, repressed हो जाएगा. Okay, we'll try to understand here what is happening in this condition. हमने क्या देखा? There is high glucose level. The concentration, I'll just write it in the proper fashion again. You can see here what is happening. The glucose concentration is higher in the cell. As a result, the adenyl cyclase, or you can say the adenylate cyclase is lower, or we can say it is, sorry, inactive, not lower. Okay, it is inactive. As a result, what is happening? The cyclic AMP concentration is lower in the cell. You can see there is always an inverse relationship between the glucose concentration and the cyclic AMP concentration. When cyclic AMP is lower in the concentration, it will not bind to cap. It will not bind to cap. Okay, वो cap protein पे bind नहीं करेगा. As a result, the cap is alone. Okay, this is alone and it cannot bind to the cap site. Cap site, that is what? This is the cap site. Okay, it is not able to bind to the cap site, and that's why there is no positive regulation, and that's why the operon is off. Right? Fine. The Lac Operon is still low. Okay, in the second condition where there is only lactose, now all of you understand. Now we are keeping a bacteria in a condition where there is only lactose. Okay, अब यहाँ पे lactose ही lactose. Okay, that means जब lactose ही है तो मतलब glucose नहीं है. Alternatively we can say ना, there is no glucose. So you can see what is happening. The glucose level is almost zero, or glucose is not there, or glucose is very low. What will happen? The adenyl cyclase will become active. Once the adenyl cyclase is active, the cyclic AMP will be made. Its concentration will increase within this bacterial cell, and it will go and bind to cap protein. वो जाके cap protein से bind हो जाएगा. Now the complex of cyclic AMP and cap can bind to the cap site, and then this cap site, this will entirely give the positive signal to RNA polymerase by bending, and now RNA polymerase can easily initiate the transcription, and the transcription of the genes will be switched on. Right? All of you definitely see here, because there is lactose, simply आपके mind में क्या click होना चाहिए? Now the gene has to be switched on. So there will be two mechanisms. पहला, you can see here, now the cap and cyclic AMP will be, this entire complex will be formed, which will give the positive effect on the operon, and the gene will be switched on. Also, because there is lactose here, what is happening? Go to the another point जो हमने दूसरा regulation देखा था. The some amount of lactose via the leaky transcription, it will enter into the cell. Right, the lactose will convert into allolactose. It is going to induce the repressor, and it will change the shape, or we can say it will block the Lac repressor. दूसरा mechanism क्या हो जाएगा? It will block the Lac repressor. Okay, this we have already talked in the another part 1 video. Okay, now the repressor cannot bind to the operator. Operator is empty. Again, the RNA polymerase can initiate the transcription, and the operon is induced. Okay, so when lactose is there, both the mechanism are going to take their function. Why? Means the first one is via the cap protein, the another one was a Lac repressor. क्या हो गया? Block हो गया. As a result, the operon will be switched on. Okay, see here what is happening here. I'll just write it here. Here we'll say a high concentration of lactose is there. Okay, indirectly what it means there is no glucose or very low concentration of glucose. What will happen here? The adenylate cyclase or adenyl cyclase is going to be active. Adenyl cyclase यहाँ क्या हो जाएगा? Active हो जाएगा. Once it is active, what it is going to do? It will increase the cyclic AMP concentration. Okay, cyclic AMP will go and you can see it will go and bind to the cap protein. This is the cyclic AMP. And this complex will go and bind to the cap site. Okay, and it is giving the positive effect on the RNA polymerase, and the genes of the Lac Operon are said to be switched on. Right? This is one mechanism by the cap protein. Lac repressor का mechanism भी simultaneously चल रहा है. Lac repressor will be blocked and it will, यहाँ पे दूसरा mechanism क्या होगा? The Lac repressor will be blocked. Okay, this will be blocked and that's why यहाँ वो bind नहीं कर पाएगा and again the transcription will go on. Okay, now we'll talk about a condition, the third condition, where both the lactose and glucose will be there in the media. Okay, now see, यहाँ पे क्या है? Because lactose is there, तो हमारे mind में क्या आना चाहिए कि genes क्या होनी चाहिए? Switched on होनी चाहिए. But we have to also take notice of this point that even the glucose is there, and glucose is the most preferential sugar for the bacteria, so it will not switch on the lactose. It is going to keep the Lac Operon in the off format. उनको वो बंद रखेगा. Why? क्योंकि glucose है. अगर आपने if the bacteria will switch on the genes of Lac Operon, तब भी वो lactose को utilize नहीं करेगा. क्यों? क्योंकि वो glucose को utilize करेगा. So that will be a wasteful event to make the enzymes responsible for lactose metabolism. Why? Because still glucose is there. So although in this case, because definitely Lac repressor यहाँ पे bind नहीं करेगा because lactose is there, but what will happen? The glucose is high. That's why adenyl cyclase will be inactive. Cyclic AMP will be low. Okay, cap will remain unbound. Cap site will be empty and it cannot initiate the RNA pol, cannot initiate the transcription. The operon will be repressed. Operon फिर भी बंद ही रहने वाला है. ठीक है, मैंने आपको बोला था, if you remember in the previous part 1, that the Lac repression का जो repression है, that repression is not very tight repression. It's a weak repression. Okay, so see here what will be the case. I'll just draw. We'll just draw it here. This is the DNA. Okay, these are our structural genes Z, Y, and A. Okay, here is the operator. Because now along with the glucose, the lactose is also there in the media. That's why here, because of the presence of lactose, the Lac repressor will be blocked. The Lac repressor will be blocked and the site is empty. Okay, so because of that, what can happen? We can understand ki what may happen ki the genes can be switched on. But we have also seen ki what is happening here. Here is a promoter and there is a cap site. Right here, because अगर इसको on रखना है, but what is happening? This is a weak repression. ये repression क्या है? Weak repression है. Okay, ये चीज weak है यहाँ पे. But cap protein is there and the cap protein यहाँ पे क्या रहेगा? It is empty. The cap site is empty. That's why यहाँ पे positive regulation नहीं हो पा रहा है. Okay, why? This is all because a preferential source glucose है. So भले ही यहाँ पे Lac repressor bound नहीं है, जिसके कारण जीन को on होना होता है, पर वो होंगे नहीं. It will not take place. Why? Because the preferential source of the glucose is there, and because of the glucose, the positive regulation, which is a strong regulation, will not be there, and glucose will show its effect, and that's why the operon will be switched off. Operon बंद ही रहेगा. Okay, you have to just remember here. Okay. Chali. So this is the last slide. We'll just summarize the effect of the lactose and the glucose. If there is lactose and glucose in the media, definitely lactose है यहाँ पे. Okay, तो media क्या हो सकता है? The operon can be switched on, but we have to also take in the account that with lactose, glucose is also there, and when glucose is also there, the operon will always be in the switched off format. ओपेरॉन बंद ही रहेगा. Why? Because this is the preferential sugar. Lactose utilize नहीं होगा until and unless glucose is there. Next condition you can see, when there is only glucose and low lactose. जब lactose नहीं है तो definitely what will happen? The lac repressor will come and bind here, and as a result the operon will be switched off. Okay, दूसरा, when there is neither glucose nor lactose, ना ही glucose है ना ही lactose है, दोनों ही नहीं है, इसके कारण क्या होगा यहाँ पे, तब भी इस operon, you can see the operon will be switched off. ओके क्यों? क्योंकि लैक्टोज नहीं है तो लैक रिप्रेसर इसको बंद ही करके रखेगा, तो द जीन्स विल बी स्विच्ड ऑफ. If there is only lactose, then only the sugar will be the lactose sugar will be utilized and the operon will be switched on. Okay, so always remember lac operon kab on hoga? When there is only lactose. Okay, remember only lactose. It can only be switched on only in a condition where there is only lactose present in the media. Okay, if this is with the combination of glucose, so भले ही lactose present है, it will not be utilized first. Okay, भले ही lac repressor यहाँ bind नहीं कर रहा है, but still what will happen? The operon will be off. Okay, so always remember operon will be on only in the condition where there is only lactose in the media. Okay everyone, so this was about the Lac Operon. We have talked about the Lac Operon in two videos. In the first video, we talked about the general what is Lac Operon, and it's the first mechanism of its regulation, that is by the Lac repressor. In today's video, we talked about the another type of regulation of Lac Operon, that is via the cap proteins. Okay, so this is the end of the Lac Operon. We will meet in the next lecture. Okay, till then have a good day everyone.